Secretions of the small intestine. Secretion from the mucosa of the small intestine contain mainly mucus, ions, and water. Intestinal secretions lubricate and protect the intestinal wall from the acidic chyme and the action of digestive enzymes. They also keep the chyme in the small intestine in a liquid form to facilitate the digestive process. Most of the secretions entering the small intestine are produced by the intestinal mucosa, but the secretions of the liver and the pancreas also enter the small intestine and play important roles in digestion. The epithelial cells in the walls of the small intestine have enzymes bound to their free surfaces that are significant in the final steps of digestion. Peptidases break the peptide bonds in proteins to form amino acids. Disaccharidases break down disaccharides such as maltose into monosaccharides such as glucose. The amino acids and the monosaccharides can be absorbed by the intestinal epithelium. Mucus is produced by duodenal glands and by goblet cells, which are dispersed throughout the epithelial lining of the entire small intestine and within intestinal glands. Hormones released from the intestinal mucosa stimulate liver and pancreatic secretions. Secretions by duodenal glands is stimulated by the vagus nerve, secretin release, and chemical or tactile irritation of the duodenal mucosa. Movement in the small intestine, mixing and propulsion of chyme are the primary mechanical events that occur in the small intestine. Peristaltic contractions proceed along the length of the intestine for variable distance and cause the chyme to move along the small intestine. Segmental contractions are propagated for only short distances and mix intestinal contents. The ileocecal sphincter at the juncture of the ileum and the large intestine remains mildly contracted most of the time, but peristaltic contraction reaching the ileocecal sphincter from the small intestine cause the sphincter to relax and allow chyme to move from the small intestine into the cecum. The ileocecal valve prevents movement from the large intestine back into the ileum. A major function of the small intestine is the absorption of nutrients. Most absorption occurs in the duodenum and yeyunum, although some absorption also occurs in the ileum. Functions of the liver. The liver performs important digestive and excretory functions, stores and processes nutrients, detoxifies harmful chemicals, and synthesizes new molecules. The liver produces and secretes about 600 to 1,000 milliliters of bile each day. Bile contains no digestive enzymes, but it plays an important role in digestion by diluting and neutralizing stomach acid and by dramatically increasing the efficiency of fat digestion and absorption. Digestive enzymes cannot act efficiently on large fat globules. Bile salts emulsify fats, breaking the fat globules into smaller droplets, much like the action of detergents in dishwater. The small droplets are more easily digested by digestive enzymes. Bile also contains excretory products such as cholesterol, fats, and bile pigments, including bilirubin, a bile pigment that results from the breakdown of hemoglobin. Gallstones may from if the amount of cholesterol secreted by the liver becomes excessive and is not able to be dissolved by the bile salts. Neural and hormonal stimuli regulate the secretion and release of bile. Bile secretion by the liver is stimulated by parasympathetic stimulation through the vagus nerve. Secretine, which is released from the duodenum, also stimulates bile secretion and release. Cholecystokinin stimulates the gallbladder to contract and release bile into the duodenum. In addition, approximately 90% of bile salts are reabsorbed in the ileum. The blood carries the bile salts back to the liver, where they stimulate additional bile salt secretion and are once again secreted into the bile. The loss of bile salts in the feces is reduced by this recycling process. The liver can remove sugar from the blood and store it in the form of glycogen. It can also store fat, vitamins, copper, and iron. This storage function is usually short-term. Foods are not always ingested in the proportion needed by the tissues. If this is the case, the liver can convert some nutrients into others. For example, if a person eats a meal that is very high in proteins, a large amount of amino acids, and only a small amount of lipids and carbohydrates are delivered to the liver. The liver can break down the amino acids and cycle many of them through metabolic pathways to produce ATP and to synthesize lipids and glucose. The liver also transforms some nutrients into more readily usable substances. Undigested fats, for example, can be combined with chlorine and phosphorus in the liver to produce phospholipids, which are essential components of cell membranes. Many undigested substances are harmful to body cells. In addition, the body itself produces many byproducts of metabolism that, if accumulated, are toxic. The liver is an important line of defense against many of those harmful substances. It detoxifies them by altering their structure, which makes their excretion easier. For example, the liver removes ammonia, a toxic byproduct of amino acid metabolism, from the circulation and converts it to urea, which is then secreted into the circulation and eliminated by the kidneys in the urine. The liver also removes other substances from the circulation and excretes them into the bile. The liver can also produce unique new compounds. Many of the blood proteins, such as albumins, fibrinogen, globulins, and clotting factors, are synthesized in the liver and realized into the circulation. Functions of the pancreas. The exocrine secretions of the pancreas include bicarbonate ions, which neutralize the acidic chyme that enters the small intestine from the stomach. The increased pH resulting from the secretion of bicarbonate ions stops pepsin digestion but provides the proper environment for the function of pancreatic enzymes. Pancreatic enzymes are also present in the exocrine secretions and are important in digesting all major classes of food. Without the enzymes produced by the pancreas, lipids, proteins, and carbohydrates cannot be adequately digested. The major proteolytic enzymes are trypsin, chymotrypsin, and carboxypeptidase. These enzymes continue the protein digestion that started in the stomach, and pancreatic amylase continues the polysaccharide digestion that began in the oral cavity. The pancreatic enzymes also include lipase, a lipid digesting enzyme, and nucleases, which are enzymes that degrade DNA and RNA to their components nucleotides. The exocrine secretory activity of the pancreas is controlled by both hormonal and neural mechanisms. Secretin initiates the release of a watery pancreatic solution that contains a large amount of bicarbonate. The primary stimulus for secretin release is the presence of acidic chyme in the duodenum. Cholecystokinin stimulates the pancreas to release an enzyme-rich solution. The primary stimulus for cholecystokinin release is the presence of fatty acids and amino acids in the duodenum. In turn, enzymes secreted by the pancreas act to digest these fatty acids and amino acids. Parasympathetic stimulation through the vagus nerves also stimulates the secretion of pancreatic juices rich in the pancreatic enzymes. Sympathetic action potentials inhibit pancreatic secretion. Functions of the large intestine. Normally, 18 to 24 hours are required for materials to pass through the large intestine, in contrast to the 3 to 5 hours required for chyme to move through the small intestine. While in the colon, chyme is converted to feces. The formation of feces involves the absorption of water and salts, the secretion of mucus, and extensive action of microorganisms. The colon stores the feces until they are eliminated by the process of defecation. Numerous microorganisms inhabit the colon. They reproduce rapidly and ultimately constitute about 30% of the dry weight of the feces. Some bacteria in the intestine synthesize vitamin K and other vitamins which are passively absorbed in the colon. Every 8 to 12 hours, large parts of the colon undergo several strong contractions called mass movements, which propel the colon content a considerable distance toward the anus. Each mass movement contraction extends over 20 or more centimeters of the large intestine, which is a much longer part of the digestive tract than that covered by a peristaltic contraction. These mass movements are very common following some meals, especially breakfast. Feces distend the rectal wall and stimulate the defecation reflex, which involves local and parasympathetic reflexes. Local reflexes cause weak contractions, whereas parasympathetic reflexes cause strong contractions and are normally responsible for most of the defecation reflex. Action potentials produced in response to the distension travel along sensory nerve fibers to the sacral region of the spinal cord, where motor action potentials are initiated that reinforce peristaltic contractions in the lower colon and the rectum. Action potentials from the spinal cord also cause the internal anal sphincter to relax. The external anal sphincter, which is composed of skeletal muscle and is under conscious cerebral control, prevents feces from moving out of the rectum and through the anal opening. If this sphincter is relaxed voluntarily, feces are expelled. The defecation reflex persists for only a few minutes and quickly subsides. Generally, the reflex is reinitiated after a period that may be as long as several hours. Mass movement in the colon are usually the reason for the reinitiation of the defecation reflex. Defecation can be initiated by voluntary actions that stimulate a defecation reflex. These actions include a large inspiration of air, followed by closure of the larynx and forceful contraction of the abdominal muscles. As a consequence, the pressure in the abdominal cavity increases and forces feces into the rectum. Stretch of the rectum initiates a defecation reflex. The increased abdominal pressure also helps push feces through the rectum. Digestion, absorption, and transport. Digestion is the breakdown of food to molecules that are small enough to be absorbed into the circulation. Mechanical digestion breaks large food particles into smaller ones. Chemical digestion involves the breaking of covalent chemical bonds in organic molecules by digestive enzymes. Carbohydrates break down into monosaccharides. Lipids break down into fatty acids and monoglycerides. And proteins break down into amino acids. Absorption. Absorption begins in the stomach, where some small lipid-soluble molecules such as alcohol and aspirin can diffuse through the stomach epithelium into the circulation. Most absorption occurs in the duodenum and jejunum, although some occurs in the ileum. Some molecules can diffuse through the intestinal wall, whereas others must be transported across the intestinal wall. Transport acquires carrier molecules and includes facilitated diffusion, co-transport, and active transport. Co-transport and active transport require, as you know, require energy to move the transported molecules across the intestinal wall. Alright guys, please subscribe to my channel, put your comments on the comment section, give me your likes, bye bye, see you.